You know, every single day, deep inside your bones, there's this incredible microscopic factory floor just humming with activity. It's pumping out millions of elite defenders for your body. So today, we're going to go inside that factory. We're going to follow the amazing journey of just one single cell as it gets forged into a powerful antibody-producing machine. Let's start right at the beginning.
So here's the fundamental challenge, right? This is the big problem your immune system has to solve. It needs to build this massive army of B cells with a huge arsenal of weapons, antibodies, that can recognize basically any foreign threat imaginable. But, and this is the really critical part, it has to make absolutely sure those weapons don't turn on your own body. It is an incredibly high-stakes balancing act between power and control.
And the best way to understand this is to think of it like a hero's journey. Seriously, our hero starts out as this humble little trainee in the bone marrow, and it has to go through these grueling trials and transformations. It faces life or death tests, and if it makes it through all of that, it finally graduates to become a mature guardian, ready to protect you. So let's follow its path.
Okay, so our story begins right here, inside the bone marrow. Our main character is a hematopoietic stem cell, which is just an amazing type of cell that has the potential to become any kind of blood cell. But today, it gets a very specific call to adventure. It's going to become a B lymphocyte.
You see, this whole development process isn't random at all. It's more like a guided tour. The bone marrow is set up with these specialized zones, or niches, and our developing B cell has to move from one niche to the next. And in each one, it interacts with different stromal cells that act like instructors, giving it the exact signals it needs to level up.
And this movement is like a highly choreographed dance. First, as a stem cell, it's kind of tethered near the bone by a signal called stem cell factor, or SCF. Then it starts to move, following this chemical breadcrumb trail called CXCL12. And finally, to become what's called a pro-B cell, it has to find a whole new set of instructors that give it this vital growth juice called interleukin-7, or IL-7. Each step pushes it further down the path, locking in its destiny to become a B cell.
All right, so its destiny is set. Now our cell faces its most important task. It has to forge its weapon. And for a B cell, that weapon is the B cell receptor. That's the antibody it's going to wear on its surface to recognize one and only one specific invader out of billions.
So how in the world does it create this one-of-a-kind weapon? Well, it uses a process that is, for all intents and purposes, a genetic lottery. It's called VDJ recombination. The cell literally takes its own DNA, snips out specific little segments, the V, D, and J pieces, and then pastes them back together in a totally random order. This genetic gamble is how your body can generate billions of different antibody shapes from a pretty limited number of genes. It's absolutely genius. And this whole assembly process happens in a very specific order. First, a D segment gets joined to a J segment. That happens early on. Then, a little later, a V segment gets attached to that DJ combo. And voila, the most important part of the antibody, the heavy chain, is complete. But, just building it isn't enough. Now it has to be tested.
And this brings our little hero to its first major checkpoint. It's forged the main part of its weapon, the heavy chain. But, does it work? Is it functional? The cell's entire existence hangs on the answer to that question.
The thing is, the cell can't just wait around to build the final light chain to see if the heavy chain is any good. So it does something really clever. It uses a temporary stand-in called the surrogate light chain. This surrogate pairs up with the brand new heavy chain to form a temporary pre-B cell receptor. And this little complex has just one job: send a signal back into the cell that basically says, "Houston, we have a winner. The heavy chain works."
And the consequences of this test? They are absolutely black and white. If that pre-BCR sends the success signal, the cell gets the green light to live. And as a reward, it gets to multiply like crazy. It also locks in that successful heavy chain, so it only makes one kind. But if there's no signal, the cell is considered a dud and is ordered to self-destruct. That's it. No second chances at this stage.
And the reward for passing is a big deal. A single successful cell will divide up to 7 times. This creates a whole clone of daughter cells that all share that exact same proven functional heavy chain. This is just incredibly efficient, right? It takes one successful roll of the genetic dice and amplifies it, creating a ton more chances to build a complete antibody in the next step.
So, our cell passed its first test. It assembles its complete receptor, but now it faces the final exam. And you could argue this one's even more important. It has to prove it's not a traitor.
Okay, so now our cell is sporting a complete IgM receptor on its surface.
It's officially an immature B cell. Its weapon is fully built. But before it can be released into the wild, it gets exposed to a whole bunch of self-molecules right there in the bone marrow. This test is called negative selection, and its only goal is to find and destroy any cell that recognizes the body's own tissues.
So what happens if its brand new weapon latches onto a self-antigen? This is the moment of truth. If a B cell fails this test, it's immediately flagged as dangerous, and one of two things is about to happen.
Now, what's so cool about this checkpoint is that unlike the first one, the system actually has this elegant backup plan. The B cell gets a second chance. It can fire up that same VDJ machinery it used before, but this time it edits the light chain. It just swaps out the genetic parts to make a brand new receptor, hoping that this version won't be self-reactive. It's this incredible attempt to salvage a cell that's almost ready to go.
But if that second chance doesn't work out, or if that initial dangerous signal is just way too strong, there's only one outcome left: elimination. The cell is ordered to undergo apoptosis, programmed cell death. This process is called clonal deletion, and it is the ultimate safety switch, making sure these dangerous, self-reactive B cells are taken off the board for good.
All right, having passed both of those grueling tests in the bone marrow, our hero B cell is now deemed safe. It basically graduates from the training academy and travels through the bloodstream to a new location for its final round of maturation, the spleen. Once it gets to the spleen, it's known as a transitional 1 or T1 B cell. From there, it moves through specific zones, maturing into a T2 B cell. And that T2 stage is key, because it's only as a T2 cell that it gets the security clearance it needs to enter the B cell follicles. Think of those as the command centers where it will finally become a fully mature defender. So what's the big difference between a T1 and a T2 cell? Well, a few crucial things change. T2 cells start putting a second kind of receptor on their surface, called IgD. They get a key survival receptor called the BATH receptor. But here's the most important part. Their whole response to an antigen signal flips. A strong signal that would have killed a T1 cell now actually sends a survival signal to a T2 cell. The recruit is finally ready for active duty.
And that brings us to this BAF signal, which is absolutely essential for survival. BAF is basically a limited resource. Think of it like a ration or a survival token that only a certain number of B cells can get. T2 cells have to compete for this BAF signal to survive and finally join the long-lived pool of mature B cells. This competition is what makes sure your body maintains a perfectly balanced number of defenders at all times.
And here they are, the final products of this whole incredible journey. Our hero cell is the one on the left, the mature follicular B cell, or B2 cell. This is the workhorse of your adaptive immune system, ready and waiting to respond to an infection. We're also showing some other specialized graduates, like B1 and marginal zone cells, which have different, more rapid response jobs in the body.
So let's just recap our hero's epic journey for a second. It started as a blank slate stem cell. It gambled with its own DNA to forge a totally unique weapon. It survived 2 incredibly intense quality control checkpoints, proving first that its weapon worked, and second, that it wasn't self-destructive. And finally, it graduated in the spleen, becoming a mature, battle-ready defender, one of millions that complete this exact same journey every single day. This whole system of checks and balances is just remarkable, and it's super effective. But it's not perfect. I mean, with millions of cells rolling off this production line every day, mistakes can and do happen. So what happens when a self-reactive cell slips through the cracks? Well, that failure, that's the origin of autoimmune disease. But that's a story for another day.